In the second part of Chapter 2, we're going to take a look more at the actual microscopes themselves and see how varying some of the different setup can actually give you distinct differences in both contrast and resolution. So if we look at sort of this idea of microscopes that are out there, and you will see that there are a couple of basic kind of setups for this stuff. Here we can see the setup for the light microscope, and this is inverted so that it matches the order of the stuff for our electron microscope. And the important thing to remember here is that lenses are actually going to be refracting or bending the light, and that's going to magnify the image of our specimen. And when we're looking at a light microscope, we use a certain set of wavelengths of that electromagnetic radiation that I talked about in the first part of this lecture for Chapter 2. For an electron microscope, you can't actually use lenses because the electrons can't go through something that's solid like that. So instead, you're going to actually use magnets to do this process here. And remember, when we look at our compound light microscope, we find that they actually have two sets of lenses that are going to be involved in the magnification process here. This lens right here is the one that's going to be condensing the light, so it's not actually doing any magnification, but it is putting a more intense beam onto your specimen. And then with your objective lens and the lens in the eyepiece, that's where we get our two sets of magnification. So I did want to distinguish between those. Same idea when you're looking at an electron microscope, and you can see that in the picture here. The electron gun is up on the top, and it's typically going to be something like a carbon fiber that you have there, and you run a massive amount of electricity into it, and the electron gun is going to shoot the electrons here, and then you use a magnet to actually condense those electron particles, just like you would be with your condenser lens over here for your light. So really kind of interesting thing to think about. And then just like we do with our lenses in the microscope, you're going to be using magnets here to magnify your electron beam right there. And here the image is actually going to end up on your eye, or you can project it. Over here, your final image is typically going to be onto a fluorescent screen. And in the old microscopes, if you look at this one over here, this little chamber here has a little screen down there, and when the electrons come off of there, they cause the screen to glow, and so they would take a picture. And some of the old school ones, you would literally hold a camera up to this window here and shoot a picture of the glowing stuff that you would see on that screen right there. So the thing I just wanted to point out here is this idea that we do use lenses in both of these setups here to get that. And remember, when we look at total magnification, it's the product of the magnification power for each of the lenses that are being used. So remember, you've got your objective lens right here that can rotate around. We use 4, 10, 40, and 100 times magnification. And your eyepiece for the microscopes is typically going to magnify things 10 times there. And with your electron microscope, you're doing your magnification of your electrons there, using your magnets to basically bend them and make the image appear larger than it actually would be. If we take a look at this image, there's a lot going on here, but the kind of interesting thing is this is the same type of specimen. This is a paramecium cell that's being viewed with different types of microscopes. We're used to seeing this kind of image over here from a bright field microscope. It's cheap, it's quick, and easy to view the specimens, but as you can see here, we do run into a little bit of resolution problem and also contrast, because those cells, like I said before, are mostly water, so they're transparent to that beam of light, and they're going to lack that contrast. So people have come up with a variety of different techniques, both with light microscopes over here and with the electron microscope, to allow you to visualize different kinds of structures in the cells that are out there. So when we look at our bright field microscope, effectively this is like you looking through a glass window, and you see the dirt on the glass, not the glass. The light is passing through the transparent thing, and in this case our cell would be the dirt on the glass that we're actually able to see, because it is bending that light, and you have a little bit of contrast there. And so I wanted to show people these different types of microscopes that are out there that we can just change the setup around to improve our contrast, change our magnification that we can use, and also get that resolution to see those structures and also to see those cells. One of the kind of interesting features when we do look at a light microscope is when that light travels from the light source and it hits the glass, that's going to cause a little bit of refraction because it's going from air to glass. And remember, when you change media, that causes your light to bend, but then it goes from the glass into the air. And this isn't really apparent at lower magnifications, but when we get up to higher and higher magnifications, that bending of the light when it hits the air, because the air is different from the glass in terms of its refractive index, that actually causes your image to become blurry and you lose some resolution. And so the solution for that is to actually take and put a little drop of what's known as immersion oil on top of your slide, and you can have a cover slip or not. When we look at our bacteria cells this semester, we're just going to put the oil directly onto the slide with the cells, and there's no cover slip, and your lens will literally be touching the oil right there. And if there's oil there, there's no air there. And kind of the cool thing is they found that certain types of oil have a very similar refractive index to your glass, and that effectively takes air out of the equation. And so now when the light passes in to your 100x objective and it gets magnified, you're not going to lose the resolution like you would in the absence of that. So really kind of a cool thing to think about. And I found this image out there, how to use microscope immersion oil, and you can see the difference between this one over here that has immersion oil, or this one over here that does not have it, and this one over here that actually is using the immersion oil. Very, very sharp, distinct features to your cells in this slice of tissue here, and you can resolve the cells in a lot of those different structures there. One of the kind of interesting features is people have found by adding an extra little piece that looks like this when you view it from above. So imagine taking this disc that has a little cutout ring around here and dropping that into the path of light. So your light comes up here, it gets condensed and it hits your specimen, and then you've got this dark field stop that is placed in the pathway that the light is traveling there. Well, what this does is it actually blocks the light that is being transmitted. But if the light is refracted or scattered by your specimen, then that light is going to be able to pass through this little ring right here, and that scattered light is the one that's going to get transmitted, and then it's going to hit your objective lens and get magnified from there. And what happens with this dark field microscope, as you can see from this image, is you actually do have a dark background right there, and your cells are actually the ones that are scattering the light, and so that allows you to see them and you get your contrast against the background. And this is really good if you think about it. If you've got a transparent cell, now you're not relying on it refracting the light in order to see against a bright background. You're actually getting the refracted light because that's the only thing that's doing it, and everything else is dark. The other thing is, if you're looking at cells that are swimming around, that's going to make it really easy to see a bright colored, say, a dot moving around on a dark background, as opposed to a pretty transparent cell against a dark background. That's going to be much harder to get your contrast to allow you to actually visualize it. So just by inserting this dark field stop, we can totally change the appearance of the image that is coming out there. One other thing that people have done that's kind of interesting also, and again, this is a figure from the book, this is what's known as a phase contrast microscope. And if you look at the difference, this is your normal bright field, and this is the one with your phase contrast microscope. So you can see they are very distinctly different images in terms of the resolution of a lot of these structures here. And when you're using a phase contrast microscope, think about those waves when you drop a pebble into some water. You're going to see repeating waves going out. Well, if your waves are in phase, they can actually reinforce each other. So remember that little peak and trough when we were looking at the wave picture at the beginning of this chapter. If the waves are in phase, they reinforce each other, and if they're out of phase, they cancel each other out. And so this is going to give us an improvement in our contrast without using any stain, because what you're doing is basically using wavelengths of light that are in phase that are adding together there. And so your specimen is going to be refracting some of the light and changing the phase of that to increase your contrast. So again, it's your standard light microscope, adding in a couple of extra pieces here. Your standard bright field microscope, you're looking at maybe $1,000, maybe $100 for a dark field stuff. With phase contrast, you're looking at $2,000 to $5,000 for those microscopes, depending on how much you want to go in for really, really good optics on that. People found that not only could they use this phase contrast microscope, but they could use something called differential interference contrast, or DIC microscopy, unfortunate acronym there. And with this one, if you look at it, you can get really high resolution three-dimensional images here. And what you do with this is, there is your light coming out of here, and you run that light through a prism that's going to split it. So you've got two separate beams of light coming in here that are hitting your specimen, and then those are going to run through another prism here and go to your eyepiece after being magnified by the objective. And so what you're doing here is kind of how they make three-dimensional movies. In the old days, they would actually take two cameras and shoot them from different angles and then overlay the film on those things to give you that three-dimensional feel to it. Same idea is going on here with this kind of microscope. You get these very good resolutions because effectively you're viewing this specimen from two angles right there just by controlling your light. Very, very good to give you these. And if you look at this specimen right here, this is actually a mold growing, and it's live. There's no stain added to this. So just by playing around with our light, we can actually get really good contrast and resolution for our cells. Now, people want to be able to look at some of the stuff inside. You can see if there's no stain inside and it's all water, you're not going to see stuff on the inside. And so people came up with a method where you can actually paint specific targets in cells. And that's where we can use fluorescence microscopy. And some cells that are out there are actually going to fluoresce naturally. And what goes on with fluorescence is you actually shine the specimen with UV light, and the fluorescent particles are going to emit visible wavelengths of light, and you can actually get multiple different colors coming out of there. And here we can see there's actually three different colors to these things that are coming out of here. So we can actually visualize the nucleus and then some elements of the cytoskeleton in green and also in the red here. They're all being painted by using different kinds of fluorescent chemicals here. And if you look at this little picture, there's your little specimen, this little spring-like thing. And you can actually take an antibody with a fluorescent molecule attached to it, and the antibody sticks to your specimen. And that way, when you shine UV light, this thing is going to give off that visible light that we can then see on our microscope. So really nice. The other really nice thing about this is not only can you get this specific part of it, but also notice the background. It's totally black, and we're only seeing these colors where the specimen is being painted with a fluorescent molecule. So you get very, very good high contrast against a dark background there when you're looking at these fluorescently labeled structures. Another version of this that people have come up with is what's known as confocal microscopy. And here, your light is actually being shot by a laser, and so you're able to very precisely shine your light on specific areas of it. And so that's where we get this confocal part of it right there. You focus this small beam of light at one depth, and then it moves over to the next spot, and then the next spot. And this is going to allow you to actually be able to visualize these really nice two-dimensional images, but at different depths. And so if you think about that, imagine taking slices through something, and then you take all those slices and you stack them up. Well, that's what we're doing. The other microscopes were visualizing just a slice through the whole piece, and here we're stacking all of those slices together to allow us to visualize the parts of what's inside of your specimen. And here you have a biofilm, so think of it just like a slimy thing, and inside of it they've actually been able to highlight these little bacteria cells that are highlighted with this false pink color there to allow you to actually reconstruct the three-dimensional structure there. And this is what's called the optical sectioning. You take sections through there and then stack them together, and it gives you some more idea about the surrounding area and the cells within there. So a lot of different techniques can be used just with a light microscope to change your resolution in your contrast. If you use an electron microscope, this has even better resolution. It can be 4,000 times better than your standard kind of light microscope. And if you looked at your naked eye, that would be 4 million times better in terms of the resolution. And remember, when we're looking at this, you've got your magnets that are focusing these electron beams, and that's why we get so much better resolution, because the wavelength of that electron is 100,000 times smaller than visible light. And so again, think about that idea of this is moving like a wave, and that allows it to change the properties a little bit. That really small wavelength is able to pass through these different really small structures, and that allows us to resolve these much smaller things. You can see there's two different images of our paramecium here. The transmission electron microscope is kind of interesting because the electrons don't penetrate through solids very well. And so when we look at this, they do thin slices of the specimen. Those are actually going to be fixed or glued together and then dehydrated, because you have to put this into a vacuum because the electrons aren't going to work well in atmosphere. They're going to bounce around because of the particles that are in there. And you basically take this thin slice right here, and you can add various stains to it if you want to. They typically use uranium atoms because they're very dense, and that's going to improve your contrast, just like staining would on a regular light microscope. But here, the electrons are getting refracted as they pass through, just like light is getting refracted when it passes through your specimen there on those things that are opaque or slightly opaque. And then you collect your image. And so you can see, rather than looking at the paramecium cell, we're looking at the interior structures in these thin slices. Your scanning electron microscope, on the other side of this, there you actually are going to paint the surface of your specimen, typically with gold or platinum, very dense kind of metals. And when I say they paint it, it is literally they atomize it, and the atoms are deposited on the surface. But we're talking about a 1 atom thick layer. So you could take a wedding ring and it would probably last for a few years in an EM lab for this scanning EM that we see right here. The electrons come on here and they're bouncing off where that metal is, and that allows us to basically detect the difference between light and dark, if you will. And again, this electron beam is going to go along here, and they're going to touch one piece at a time and then collect that image off to the side there on that glowing screen right there. So very different. This is going to give you a 3-dimensional image of the surface. This is going to give you a 2-dimensional image of the interior of your specimen there. And we can go even further than this. When you look at what's known as probe microscopy, this little cartoon I found out there is really telling you what's going on. You have a probe, think of it kind of like a seesaw, that is reaching out and touching the atoms in your specimen, and that seesaw is going to bounce up and down as it feels the end of this. And on the other side of this, you're going to have a laser that is going to detect when your seesaw moves up and down right there. So you are literally feeling the force of the atoms. And here we can actually get down to the level of the molecules and see the actual fiber. And here you're actually looking at an image of the surface of gold, and you're seeing the individual atoms of gold that are arranged in these structures right there. And so really kind of interesting to think about that we can actually feel the atoms and these pieces of these different specimens that we want to be able to visualize with this probe microscopy. And just sort of a summary to get you all this stuff, when you're looking at your light microscope, notice the best magnification we can get, because we run into problems with our light waves there, is we can magnify these specimens 2,000 times and resolve to under 1 micrometer, down to about 200 nanometers. Great for looking at live cells, even better for looking at stained cells, because again, they don't refract light effectively there, but they do lack that contrast. With our electron microscope, depending on the type, you can get different types of magnification, but much higher magnification compared to your light waves, and the maximum resolution is going to be even smaller. And I'm looking at this. You can see that 10 nanometers, so we can get down to much smaller sized particles can be distinguished from each other. Again, 2-dimensional internal structures with the transmission and three-dimensional with the scan. You can think 3D, you scan the outside of it. With your probe microscopy, we're talking about millions of times of magnification down there. And if you look at this, we can go all the way down to a fraction of a micrometer there because you're actually feeling the surface of those objects.